Hi all, in this video we're going to see about neuroglia. So this question has been asked previously as a short essay, so we'll see what neuroglia is. So neuroglia or glial cells are basically non-neuronal cells that are present in the nervous system that support, protect and maintain the environment around the neurons. So you can see that this yellow color structures are the glial cells that help and support the neurons that are present in the central nervous system. So, the important properties of neuroglia are they do not conduct the action potential, they can proliferate in adulthood, especially after injury, and they are involved in the neural support, immune defense, nutritive roles, as well as myelin formation. So, now we'll see the classification of the glial cells. So, we can classify them into microglia and macroglia based on their size. Microglia means obviously small glial cells. Again, they are non-neural from monocytic lineage. So obviously what might be its function, because it's from a monocytic lineage, which means they act as the macrophages of CNS. So it helps to remove the cellular debris after injury or infection, like in multiple sclerosis, Parkinson's, Alzheimer's, and they also enlarge and activate during damage. So their main function is this phagocytic role. So now we'll see about the macroglia cells. So obviously they are a bit more bigger. They include the astrocytes, the oligodendrocytes, the Schwann cells, as well as the ependymal cells. So now we'll see about each one by one. First and the most important one is the astrocytes, because it's the most abundant glial cells. You know the term astro means star, so it's a star like in shape, and they connect the neurons and the capillaries. It's of two types. The fibrous astrocyte is basically present in the white matter, and the protoplasmic astrocyte is basically found in the gray matter. So what are the functions of astrocyte? They form the blood-brain barrier by inducing the tight junctions. They envelop the synapses so as to produce an electrical insulation. They regulate potassium, glutamate and GABA concentration and provide the nutritional and structural support to the neurons. They also respond to the extracellular potassium, but they do not generate action potential. So astrocytes have got multiple roles, especially in case of blood-brain barrier. They are electrolyte neurotransmitter relationships, nutritional as well as structural support. So in this image you can see that it is connecting. This is a fibrous astrocyte, which with the help of the end foot is connecting the capillary and the neuron. The next class is the oligodendrocytes. So oligodendrocytes are mainly found in the CNS, and it is actually the cells that produce myelin sheath in the CNS, in the central nervous system. So it helps in the saltatory conduction. The myelin sheath is required for saltatory conduction. So indirectly, the oligodendrocyte will help in saltatory conduction. And we know that in the peripheral nervous system, it is actually the Schwann cells that help in production of myelin sheath. So what's the difference between this and the Schwann cells? The first difference is that oligodendrocytes are present in the CNS and it can myelinate multiple axons at a time. See, like this. But Schwann cells, they are present in the peripheral nervous system. It can only myelinate one axon segment at a time. So that is a major difference between oligodendrocytes and Schwann cells. So we'll see more about Schwann cells. See, you can see that each Schwann cell is for a single segment of the axon. So it wraps a single axon segment. They form myelin sheath by rotating around the axon. See, that is how the Schwann cells produce myelin sheath around this nerve fibers in the peripheral nervous system. And we have the nodes of Ranvier, which are actually gaps that are present between the two segments, and they help in saltatory conduction, that is fast signal conduction. And the last type is ependymal cells, which are basically present in the ventricular system of the brain. You know, it is involved in the production of the cerebrospinal fluid or CSF secretion. So we have specialized ependymal cells present in the choroid plexus that help in CSF secretion. They also have a barrier function which permits diffusion of many substances between the brain extracellular fluid as well as CSF. So thus we've seen the microglia and the macroglia. So what are the different clinical correlations? So if there's a destruction of the CNS myelin, what will happen? It can lead to diseases like multiple sclerosis. If there's a mutation in this myelin gene, it can cause Charcot-Marie-Tooth syndrome. And there are other syndromes like Guillain-Barré syndrome, which is an attack on the myelin proteins present in the peripheral nervous system. So there are many syndromes that can occur if this myelination, which is produced by these macroglial cells, go wrong. Another important clinical application is you can have gliomas. That means you can have tumors of these glial cells. See, we know that neurons cannot multiply, but glial cells can. So after an injury, there can be glial proliferations which can cause gliomas, which are common brain tumors. So that is another important applied aspect of neuroglia. So thus, for neuroglia, we've seen the definition, what neuroglia is, what is the classification. We said it can be classified into microglia and macroglia. Macroglia can again be classified into astrocytes, oligodendrocytes, Schwann cells, and ependymal cells. We've seen the function of each as well as the applied aspect. So I hope this was useful for you. Thank you.